There's someone in your life who just turned 60 and something doesn't add up. Their skin looks mid-30s. Their energy, their posture, the way they move through a room. You do the math twice. Curious. Then there's someone else, maybe 10 years younger, who looks weathered and exhausted and older than they should. Same calendar age bracket. Completely different biological reality. This isn't luck. It isn't vanity. And it has almost nothing to do with genetics in the way most people think. What you're witnessing is the visible surface of a process scientists have spent 2 decades finally beginning to crack open. And what they've found is astonishing. Surprised? Aging isn't a slow, inevitable march forward on a fixed schedule. It's a dynamic, measurable, and partially controllable biological process. The gap between looking 60 and looking 35 at the same chronological age is real. It has mechanisms, and those mechanisms are things you can actually influence. Today we're going deep. We're going to talk about what biological age actually is and why it's completely different from the number on your driver's license. We'll look at 3 specific cellular mechanisms that control how fast you age, and why some people's biology runs decades behind the calendar. And we'll talk about what the research actually says you can do about it. Stay with me, because the last piece of this is the one that surprised even the scientists who discovered it. Start with this. Your chronological age is simply how many years have passed since you were born. It's a calendar measurement. Your biological age is something entirely different. It's a measure of how worn down your cells and tissues actually are, how well your DNA is being maintained, how efficiently your cellular machinery is running. And these 2 numbers, for most people, are not the same. According to a landmark study published in the Proceedings of the National Academy of Sciences, researchers at Duke University tracked nearly 1,000 people from birth to their late 30s and found that their biological aging rates varied enormously. Some 38-year-olds had the cellular profile of a 28-year-old. Others looked biologically more like someone in their early 60s. Same birthday, completely different bodies at the cellular level. Lead researcher Terry Moffat described watching some people age 3 biological years for every 1 calendar year, while others barely aged at all. 3 to 1. That gap exists right now, across the people in your life, and across different versions of the future you. So what's actually happening inside the people who seem to be aging in slow motion? To understand that, we need to zoom into something that's happening inside your cells right now. Picture your DNA as 46 incredibly long instruction manuals, the complete blueprint for everything your body does. At the very tips of every chromosome, capping the ends of each of those manuals, there are protective structures called telomeres. Telomeres work exactly like the plastic tips on shoelaces. You know how a new shoelace has that hard aglet at the end, keeping the fabric from unraveling? That's precisely what telomeres do for your chromosomes. They cap the ends and prevent your genetic information from deteriorating. But here's the problem. Every single time your cell divides, those caps get a little shorter. A little more protective material is lost. And when a telomere gets short enough, the cell can no longer safely divide. The Nobel Prize-winning research behind this discovery came from Elizabeth Blackburn, Carol Greider, and Jack Shostak in 2009. But here's what most people don't know. The rate at which your telomeres shorten is not fixed. It varies dramatically based on things happening in your body right now. A 2004 study by Alyssa Eppel and Elizabeth Blackburn at UC San Francisco found that women under chronic psychological stress, specifically those caring for chronically ill children, had telomeres equivalent to someone a decade older. Chronic stress was accelerating cellular aging at a directly measurable rate. But the reverse is equally true. Exercise, specifically sustained aerobic exercise, has been shown to upregulate telomerase, the enzyme that rebuilds telomere length. A study published in the British Medical Journal found that regular vigorous exercisers had telomeres equivalent to someone roughly 9 years biologically younger than sedentary individuals of the same age. 9 years. Not from a drug, not from a procedure, from consistent movement. Telomeres, though, are only part of the story. And in the last decade, a second concept has moved to the center of aging science, one that changes how we think about the whole process. It's called the epigenetic clock, and once you understand it, the biology of aging will never look the same. Your DNA doesn't just sit there. It's constantly being read and regulated by a second layer of information called the epigenome. Think of your DNA as the text of a massive book the actual words on every page. The epigenome is all the highlighting, the dog-eared pages, the sticky notes in the margins. It doesn't change the words themselves, but it controls which chapters get read, which sentences get emphasized, and which passages get skipped. And this annotation system changes throughout your life in remarkably predictable patterns. In 2013, computational biologist Steve Horvath at UCLA published what many consider the most significant paper in aging research in 50 years. He analyzed chemical tags on DNA called methylation marks across dozens of tissue types and hundreds of age groups. And he found something extraordinary. These methylation patterns change in a clock-like, highly predictable manner across a human lifetime. He developed what's now called the Horvath clock, a model that can look at the methylation pattern in a DNA sample and accurately predict your biological age to within about 3.5 years. Some people's epigenetic clocks run fast. Their biology is aging ahead of their calendar. Others run slow. The 60-year-old who looks 35 almost certainly has an epigenetic clock reading somewhere in the mid-40s. And here's the part that should genuinely excite you. These methylation patterns appear to be responsive to how you're living. They're not locked in. Dr. David Sinclair at Harvard Medical School published work in 2020 showing it was possible to actually reset the epigenetic clock in mice, not just slow it, but reverse it. That was in mice, and the leap to human therapeutics is real and significant. But the proof of concept was extraordinary. The sticky notes on your DNA can, at least partially, be rewritten. Now let's talk about the third mechanism, and honestly, the most underappreciated one in popular conversations about aging. It's called cellular senescence, and it might be the most actionable piece of this whole picture. When a cell reaches the end of its replicative life, or sustains damage it can't repair, it's supposed to die cleanly through a process called apoptosis, orderly, efficient, no mess left behind. But sometimes a cell doesn't die. Instead, it enters this zombie-like state. It stops dividing. It doesn't die, and it starts secreting inflammatory signals into the surrounding tissue. Scientists call this the senescence-associated secretory phenotype, or SASB. And the accumulation of these zombie cells over time is now understood to be one of the primary drivers of the tissue deterioration we associate with aging. Here's the analogy that makes it click. Imagine a city where, when a building is damaged beyond repair, instead of being demolished and cleared, it's just left standing. And it starts leaking toxic fumes into the neighborhood. Other buildings nearby start deteriorating. The whole area ages badly. That's what a senescent cell does. It poisons the tissue around it with chronic inflammatory signals, dragging neighboring healthy cells into dysfunction. A 2018 paper in Nature Medicine by James Kirkland and colleagues at the Mayo Clinic demonstrated that transplanting just a small number of senescent cells into young, healthy mice caused those young mice to rapidly develop age-related dysfunction. Conversely, selectively clearing senescent cells improved physical function and restored health markers. The cells were doing enormous biological damage, out of proportion to their numbers. Human clinical trials of senolytic compounds, drugs that selectively clear senescent cells, are now underway at several major research centers. The results are preliminary, but the key thing about senescent cell accumulation that you can act on right now is this. Its biggest driver isn't aging itself. It's chronic low-grade inflammation, what researchers have started calling inflammaging. And inflammaging is driven by exactly the same factors that accelerate telomere shortening and epigenetic clock advancement. Are you seeing the pattern? 3 completely different biological mechanisms, studied by different teams at different institutions using different methods. And when you trace what drives all 3 of them, you keep arriving at the same short list. Chronic inflammation, chronic stress, metabolic dysfunction, sleep deprivation. These aren't separate risk factors for 3 separate problems. They're the same upstream causes producing 3 convergent downstream outcomes. Fix the upstream, and the downstream improves across all 3 simultaneously. Before we go further, I want to address the genetics question directly, because most people assume it's the dominant factor. The research says otherwise, and the margin is not close. A 2018 study published in the journal Genetics analyzed data from 54 million people and found that the heritability of longevity is closer to 7%, not the 25 to 30% that was previously assumed. 7%. The vast majority of variation in how people age is attributable to non-genetic factors. Your genes load the gun. Everything else pulls the trigger, or doesn't. So what does the evidence actually say about the levers you can pull? Let me be honest here. The popular conversation about lifestyle and aging gets lost in the details, the specific superfoods, the exact supplement stacks, the precise fasting windows. The research suggests the big picture is simpler than that, and that the fundamentals outweigh any optimization at the margins. Sleep is the one I want to start with, because it's the most underweight factor in how people think about aging, and arguably the most powerful. We tend to think of sleep as passive, as the absence of activity. The research reveals something completely different. During deep sleep, your brain activates a system called the glymphatic network, a series of channels that essentially flushes out metabolic waste products that accumulate during waking hours, including, critically, beta amyloid and tau proteins, the very proteins associated with Alzheimer's disease. A 2019 study published in Science showed that a single night of sleep deprivation led to significant tau protein accumulation in the human brain. One night. Chronic sleep disruption is associated with accelerated telomere shortening, elevated inflammatory markers, impaired glucose metabolism, and disrupted DNA repair, essentially hitting every single aging mechanism we've discussed, all at once. Matthew Walker at UC Berkeley, whose research has done more than almost anything to reframe how we think about sleep, has described chronic sleep deprivation as the most powerful known accelerant of biological aging. Not a contributor to it, the most powerful accelerant. Exercise is the other foundational intervention, and its effects on biological aging are broader than most people realize. It's not just about cardiovascular health or body composition. Regular physical activity upregulates mitochondrial biogenesis, the creation of new cellular power plants. It reduces inflammatory markers. It improves insulin sensitivity, which is one of the central metabolic drivers of biological aging. It promotes the clearance of senescent cells. It improves sleep quality, and it has direct effects on the epigenetic clock. Multiple studies have found that consistent exercisers carry methylation patterns associated with biological ages significantly younger than their sedentary peers. The research strongly suggests that any consistent movement is dramatically better than none, and that the difference between doing something and doing nothing dwarfs the difference between any two specific exercise protocols. Resistance training preserves muscle mass and metabolic rate. Sustained aerobic work at moderate intensity is particularly effective for mitochondrial health. Brief high-intensity intervals trigger cellular stress responses that strengthen repair mechanisms. The ideal probably involves some of each, but the most important variable is consistency over years, not optimization within weeks. Here's the counterintuitive finding that surprised me most when I went deep into this research. You might assume the people who age slowest are the ones who've been hyper-vigilant about their health, obsessively optimizing every variable. But a 2021 study from the Stanford Center on Longevity found that a significant predictor of healthy aging wasn't health-related anxiety or rigorous protocol following. It was a sense of purpose and meaning. People who reported high levels of purpose, who felt engaged with their work and their community, who had strong social bonds, showed significantly better biomarkers of biological aging than those who didn't, even after controlling for diet, exercise, and all other lifestyle factors. This connects to hard biology. Chronic loneliness triggers the same inflammatory cascades as physical injury. A meta-analysis published in Perspectives on Psychological Science found that social isolation was associated with a 26% increased risk of premature mortality, comparable in effect size to smoking up to 15 cigarettes per day. The biology of loneliness is the biology of accelerated aging. The depth of your relationships, the degree to which you feel your life has direction and meaning, appears in the data as a direct contributor to your biological age trajectory. That's not soft science. That's measurable cellular impact. Let me be honest about the limits of what we know, because intellectual honesty here matters more than a clean narrative. The mechanisms, telomeres, the epigenetic clock, senescent cells, the inflammatory pathways, these are well-established science. The lifestyle factors I've described are supported by significant, replicated evidence. But the idea that we can dramatically and intentionally extend human health span through targeted interventions, is still being actively tested. The mouse data is compelling. The human data is encouraging but early. Anyone promising certainty about radical life extension is ahead of what the evidence actually supports. What we can say confidently is this: the gap between biological age and chronological age is real, measurable, and significantly shaped by upstream factors you have genuine control over. Sleep quality, chronic inflammation, metabolic health, consistent exercise, stress, not just its presence, but whether it's chronic and unrelenting, social connection, and a sense of purpose. These aren't separate, parallel factors. They're interconnected levers on the same underlying biology, and they compound over decades. The 60-year-old who looks 35 isn't mysterious. She isn't doing something exotic or expensive. Her epigenetic clock reads a decade younger because of how she's lived, not a single dramatic intervention, but a consistent pattern over time. Her telomeres are longer not because she won a genetic lottery, but because the biological inputs she's been feeding her cells have kept them that way. Her tissues are less inflamed, her senescent cell burden is lower, and her cellular repair machinery is running closer to spec. The biology is responding to her life. Understanding the mechanism changes something. It's one thing to hear that you should sleep more and move your body and manage your stress. It's another thing entirely to know that right now, the methylation marks on your DNA are being shaped by how you're living, that your telomeres are responding in real time to the cortisol in your bloodstream, that every night of good sleep is your glymphatic system running maintenance that nothing else can replicate. You're not a passive observer of your own aging. The biology has always been responding to you. Now you just know what it's responding to. If you want to go deeper on where this science is actually headed, the emerging research on senolytics, the epigenetic reprogramming work that's moving from mice into early human trials, and what the Blue Zones data tells us about the biology of purpose, subscribe, because that's exactly where we're going next. The next video looks at the people who aren't just aging slowly. They appear to be aging in reverse, and what that tells us about how far this science can actually go.